Hello my friends, Assalamu alaikum and welcome back. Friends, in the previous lecture we discussed about the structure of the microvilli and stereocilia. You can go and watch that lecture. As you know, we are discussing about the features of the apical surface of the epithelial cells, right? Now I had told you in the apical surface of the epithelial cells in our body there can be microvilli, there can be stereocilia, and in certain points in our body like respiratory tract there can be cilia, right? So we have discussed about the structure of microvilli and stereocilia. Now we are going to discuss about the structure of the cilia in detail, right? Let's go for it. Friends, first of all, let me make here is a cell, right? The big cell I will draw, right? So let us see about the inside surface, inside structure of the cilia here. These were the small pillars, but this is a big pillar, and these are the immovable pillars. This is a motile pillar. It shows movement, right? So first of all, let me write here what are cilia. Cilia are also finger-like projections, but longer than microvilli and stereocilia. So they are also finger-like projections of cell membrane in epithelial tissue, right? Their length can be from 5 to 10 micrometer, but you can see here in case of microvilli, I had told you that their length can be 1 micrometer. So look at the difference between both. Then we had told you that the microvilli and the stereocilia are immovable, but the cilia are motile in nature. It means that they show movement. So the movement will be in this manner, right? So they show movements like this. So mostly they are present in our respiratory tract to clear the respiratory tract. So they also provide certain like immunity, right, for us. So they are motile in nature. They show movement, right, like this movement. So let us see about the structure. First of all, if you want to know very basic about the structure of the cilia, so two important things are very important. And what are those important things? The first, there is a basement for it, and then there is the pillar, right? So the basement is called the basal body, and I will discuss about the structure of basal body. And in the length of it longitudinally there are exonym. So I will discuss about the exonym structure and the basal body. So for the arrangement in the books they have written that the basal body, we write the formula as 9 plus 0. And what is 9 plus 0? I will tell you. And for exonym we write 9 plus 2. What is 9 plus 2? We will see in the right now, right? So let's first of all discuss about the basal body. So we provide a very strong foundation for such type of pillar, right, which moves, which show movement. You know, in Japan there are lots of buildings, right? So they have provided the foundations, and in those foundations they have put the springs. And during earthquakes, what will happen? The building will move in this and this manner, but it will not fall down. Why? Because they have provided strong pillars in spring-like manner for these large buildings, so that these buildings are prevented from falling down. Now same is the case here. If this cilia is moving, right, so there should be a very strong base, a very strong foundation for this cilia, so that this cilia could not be broken down and could not be fall down, right? So let's first of all discuss about the basal body. In the basal body, right, so let's see about the basal body, the structure of the basal body. So in the basal body, right, there are proteins which are arranged in such a manner, right, that they are arranged as the arrangement that you have seen in the centriole. So which type of arrangement is that? There are three groups of proteins, right? How many groups of proteins? There are three groups of proteins. So 1, 2, 3, 4, 5, 6, 7, 8, 9, right? And then there is the other one. I will draw with different color because it is very important to draw them with different colors. Right? So these are the components. Now let me make them very simple. These are three tubes, right? I will show you another picture also. Here are the three tubes. Now look carefully. Look at here. This is one tube, right? This is tubulin protein. This is the other tube, tubulin protein, right? And this is the red one. Very simple, right? So how many proteins have been united? There are 3 tubulin proteins. So they are making a triplet, right? They are making triplet, and all these triplets are connected to a central hub, right? They are connected to a central hub. This is also a protein, right? So like this. Like this, right? And they make a circular arrangement here in the base of the pillar. So that's why we call it the basal body. If you want to make it more clear, so it will look like this, right? So proteins will arise from the cytoplasm, right? And they will go up and up to the base of the cilia, right? So what are these proteins? These are tubulin proteins. For example, this is tubulin protein A, this is tubulin protein B, and this is C. So what are these? These are tubulin proteins, right? And all these proteins are connected to a central hub. Sorry, this is a central hub, and these are the spokes which connect these components of these tubulin proteins to the central hub. So there is a very strong foundation in the cytoplasm for this cilia. Now look carefully. There is nothing in the center, but around that center there are three groups of tubulin proteins arranged, which are A, B and C. How many? 1, 2, 3. 9 multiplied by 3, which equals 27. So 27 protein filaments, right, will arise from cytoplasm, and they will come upward and they will make the basal body, which will give strong foundation to the core of the cilia, which is called the exonym, right? So we see here there are three arrangements. So 1, 2, 3, 4, 5, 6, 7, 8, 9. So that's why we say there are 9 triplets, but nothing in the center. That's why we call it 9 plus 0, right? 9 plus 0. Now look, when we go up, this was a section which we made at the basal body. Now when we go up and we see here and we make a section here, what do we see? We see 9 doublets with central 2 molecules, right? What will happen? That one of these will fuse together and they will make one doublet in the center. So this is the doublet in the center, right? But around this central doublet, what is there? Look carefully. There is a protein coat which is called central sheet, right? Now I am going to discuss about the structure of the exonym. Look carefully. There is proteins around the central 2 doublets, which are called the exonym, and these doublets are also connected with each other. So these doublets are the long protein filaments, nothing else. Don't forget that these are the protein filaments, my friends, right? And they come from downward to upward and like this, and they are surrounded by this sheet which is called the this is called the central sheet, which is around these doublets like this, right? Now if you want to complete it, they will become like this. So central sheet like this. right? So this is the central sheet, and in this central sheet what has came up, there are the doublet proteins, and they are also tubulin proteins, nothing else, right? Then around this two central proteins and around this central sheet, there are arrangement of doublets, 9 doublets. Doublets means from these 3, there 2 will go up but 1 will remain down, right? So from these 3 triplets, 2 of them will go up, which will make doublets, and one will remain down. So look carefully. Again we will draw here that structure like this. So let us make here one which is little bit smaller and the other unit is little bit larger. So this small unit is called A unit and this larger unit is called B unit. You just remember it, right? Again here is A unit and here is the B unit, right? So how many doublets we will make? We will make 9 doublets. So 1 2 3 4 5 6 7 8 9. So here again one A, and this is little bit larger B, right? Then again here A component, then again here B component. You just remember please, A component and B component. So 1 2 3 4 5, B component, A component 6, then 7 8 and 9, right? And this B component and B component, right? So how many doublets have came up? Only 2 doublets, only 9 doublets, right? Doublets means what? Doublets means 2 2, and triplets means 3 3 tubulin protein filaments, right? And what is this structure really? If you want to know, this structure will be like this, right? So this is the black one, which is the A component, and this is the red one, which is the B component. So all these fibers will, like these markers which I am holding in my hand, these are the fibers, will go up 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 and up, right? So this makes, right, this is the exonym. In the structure of exonym, what do you see? Look, I will draw this one also and this one also, right? And again this one. So look, very nice structure they represent like this. So this is the A component, the red one is the B component, which is little bit larger. This is the A component and then the B component, and these doublets are coming up, right? And in the center there is the two singlets, and around that there is a central sheet. So let me write it here. Then very importantly, look carefully, I will remove this black color there. Look, from these doublets, listen to me my friends, from the A component there are certain proteins which arise and connect with B component like this. Please remember, from A component proteins arise and they connect to B component in all, and they hold them very firmly together. So these are called the, look, like this, right? From A component and enters into the B component, right? These which hold the A component of one doublet to the B component of the other doublet, this is called the Nasal protein, right? Very short protein which is called Nasal protein. And another important thing, what happens, look carefully, look here, this is the A component, look, and what is here, this is the B component, right? Then again here is the A component and this is the B component. Now look, from the A component protein arise and hold that one, this is the Nasal protein, and another protein which arises from the A component and give a hug to the B component of that other doublet. This is called another group of proteins, and these are called the Dynein arms. What are these? These are the Dynein arms, right? And these are the arms which hold the other components, right, during the movement of the cilia, and they provide movement, they provide help in the movement of the cilia. So what are these, right? What are these? These are the Dynein arms which arise from A component and hugs the B component of the other doublet like this. So these are the Dynein arms. Please remember all these. So these are the Dynein arms like this. Very simple. Okay, so this is the central sheet, right? These are the two singlets which come to the center, in the center. Then around these two singlets, what is there? The central sheet is there. And look carefully, the central sheet is connected to these doublets, right? From the A component there are proteins which come and connect to the central sheet, right, like this. These are called the radial spokes. What are these? These are the radial spokes like this. Clear? These spokes are again there. So in a very simplified way, let us write all of these. What are these? These are these green are the Dynein arms. Dynein arms, right? And what is this red one? This is the B tubule, right? This black one, A tubule, right? And then the nexine protein. And what are these? These are the radial spokes. Radial spokes are there. Then this red one is the central sheet. Central sheet, right? And these are the two singlets. Singlets means 1 1, and these two are the doublets, the A and B tubule, the doublets. And here the A, B and C tubule, A, B and C tubule, they make the triplets, right? And here you can see there are the arrangement of 9 plus 2. How? Look here, this is 9 plus 3, 9 plus 0. 9 means the 9 triplets, but in the center they have nothing, right? But here you can see the doublets, and around it, look carefully, in the center there are singlets, and around these two singlets there are doublets. And how many doublets? There are 1, 2, 3, 4, 5, 6, 7, 8, 9. So 9 plus how many singlets are there in the center? There are 2. So 9 plus 2 for the exonym, which I told you here, 9 plus 2 arrangement. So 9 plus 0 arrangement for the basal body, 9 plus 2 arrangement for the exonym. Then in the basal body, you know, there are triplets, right? So A, B, C tubulin proteins are coming, but in the doublets there are only A and B proteins. A and B, A is smaller and B is a little bit larger. So this is the arrangement of the fibers or filaments in the cilia, and they are showing movement, right? And these are the Dynein arms which help them to move right and left movement they show. So friends, here we stop our lecture about the cilia, and we have discussed about the structure of cilia, the microvilli and the stereocilia. So these were the features in the apical surface of the epithelial tissues in the bodies of animals and in the bodies of us, friends, right? Now we discussed about the structures which are present in the basal part, we discussed about the structure which are present in the lateral part, and we discussed about the structures which are present in the apical part or apical surface of the epithelial tissues. In the next lecture I will discuss about the functions of these epithelial tissues, and then we will go and we will discuss about the types of the glands. You know, I told you that epithelial tissues are present in our body as glandular cells. So, and glandular tissues, we will discuss about the types of glands also in the next lectures. So guys, for now it's enough. See you guys in the next lecture. Till then, Allah Hafiz.